Welcome back, everybody. Chapter 3, Cholesterol Metabolism. We'll try and get through this whole thing in one video, but we might have to break it up. It just depends on how long it takes us to get through all of this. So, the lecture objectives. We're going to recognize the biomedical importance of cholesterol metabolism, identify and construct the basic ring structure of sterols. You might remember sterols from Biochem 1. We talked briefly about that. Know the dietary and physiological sources of cholesterol. The dietary sources, cholesterol is going to come exclusively from animal products. Know the normal average blood levels of cholesterol, average production by liver, and average daily dietary intake of cholesterol. Describe the cholesterol packaging and transportation mechanisms in the body. Demonstrate cholesterol biosynthesis and its metabolism. Explain the regulation of cholesterol biosynthesis. Describe biosynthesis of bile acids from cholesterol and its metabolism with reference to the synthesis of conjugated bile acids and their functions in fat metabolism. So bile acids, this will be talking about the gallbladder and how the gallbladder produces specific acids from cholesterol to actually be used to be injected into the small intestine to break down fats to allow us to absorb those fats and digest them. Okay, so biomedical importance of cholesterol. Cholesterol is an essential component of membranes and outer layers of plasma lipoproteins and blood lipoproteins. The storage form of cholesterol is cholesterol esters. They are major constituents of gallstones. Gallstones, also known as cholecystitis, is going to be the stone formation that we get in the gallbladder, and sometimes they typically will require some sort of surgery to remove them. They can be very, very painful. We'll learn more about that later on down the road, about gallstones and the gallbladder and kind of that presentation. You'll learn that in other courses. So the precursor of bile acids, cholesterol is, steroids, vitamin D, etc. Several pathologic processes may produce atherosclerosis. Atherosclerosis is another word. This is hardening of vital arteries. So this is like a fatty buildup. It's a fatty buildup of the cholesterol on the arteries, and it slowly clogs them, and also the arteries lose their ability to kind of stretch and move with the pressure of the blood. So you'll get stuff like high blood pressure, and eventually you can actually have a thrombus develop. A thrombus is a blood clot within a specific area. You'll learn more about that in general pathology. I believe try 3 or try 4. And you will learn about that whole process of atherosclerosis, how it happens, why it happens, and also what can happen as a result of it. Okay, so we have the structure of cholesterol. We can see it's a multi-ring structure coming from the diet as well as the biosynthesis. You can have the biosynthesis of it, just like we talked about in the previous chapter. Acetyl-CoAs being put together to eventually form a chain long enough, and the chain forms the specific rings and gives us cholesterol. Cholesterol will then be used in the body for different membranes, for blood lipoproteins, steroid hormones, I think like testosterone, estrogen, progesterone, those sort of steroid hormones, as well as thyroid hormones. What else? There's a couple of other hormones as well that are labeled under those steroid hormones, and also bile salts, which are necessary for digestion of fats. Okay, so we have the basic ring structure of our sterols and our cholesterol. Here's our cholesterol structure. And then our phenanthrene structure. Let me double check. Okay, so phenanthrene is very much like cholesterol as well, and it's used to make bile salts or make cholesterol and well as steroids. So one of the most common ways that it can actually enter your body is through breathing contaminated air. It can get in your lungs when you breathe it. So that's interesting. So it's kind of it's a chemical as well as a different type of structure that's naturally found in our body. It's kind of like, just like anything, too much of anything is going to be bad. So we're going to use some of it to produce cholesterol. But again, if we have too much cholesterol in our bodies, we can have some not so nice things happen. Sources of cholesterol. So cholesterol is present in tissues and also in plasma lipoproteins. All tissues containing nucleus can biosynthesize cholesterol. So if we stop and we think about that, there are going to be certain cells that don't have nuclei that cannot biosynthesize cholesterol. One of the main ones that comes out of the top of my brain is our red blood cells. Red blood cells do not have nuclei, and so they cannot synthesize their own cholesterol for their membranes and for whatever that they would need. So the gene that expresses the rate-limiting enzyme in the cholesterol biosynthetic pathway and regulates its biosynthesis is localized in the nucleus. So the rate-limiting enzyme for cholesterol biosynthesis is localized in the nucleus. The endoplasmic reticulum and cytosolic fraction contain other required biomolecules for its biosynthesis, of which acetyl-CoA and the coenzyme NADPH are both important. Are both going to be important in that process. It is present as either free cholesterol or as a cholesterol ester. The cholesterol ester is the storage form in most tissues. Remember ester? We talked about ester bonds. Ester bonds they're a type of bond that you see often. Let me actually pull up a good picture for that. So we have our carbon and our OH, and then the ester bond links to that oxygen combining. Between the two, the OH combining gives us that ester bond. The starting molecule for cholesterol biosynthesis is acetyl-CoA, and it is typically a product of animal metabolism. So the cholesterol is a product of animal metabolism, and thus is present in foods of animal origin, such as egg yolks, meat, liver, and brain. So in our diet, we'll find it in egg yolks, primarily those red meats, and it is not synthesized in plants. We can also get it from reabsorbed bile salts, so those chemicals that our body is going to put out to help us digest those fats. Our body is going to recycle those and continue to kind of process them through and like excrete them, reabsorb them, remake the bile salts, or remake the bile acid, put it back into the small intestine, digest more fats, and so on and so forth, doing that process. We do lose some of it through the feces, but a good majority of it will be reabsorbed. So the dietary cholesterol input is about 300 milligrams per day. This is also something important to note. If anybody is living, like in the future when you have patients and stuff, if anybody is living a very heavily or primarily plant-based diet, especially when somebody is practicing strict like veganism, a lot of times they can have digestive type issues due to not having the cholesterol in their system. They can also have a lot of other issues if they are not properly supplementing their diet and picking the specific supplements and specific foods that they need to get all of the nutrients that they need to on a daily basis. So keep that in mind if you do have vegan patients in the future, that there are going to be times where you might have to have these sort of conversations talking about the importance of going to having a proper nutrition plan and going maybe to a nutritionist or a dietitian to help them put that together and see what that looks like if they want to continue to live that vegan lifestyle. Because in today's world, with all the supplements and everything, it's very much doable, but you just have to have the proper nutrition in order to do it. Okay, so then body synthesis in most cells of the body. So that's from the diet. We also have the synthesizing of it. So low dietary sources of sterols stimulate endogenous synthesis of cholesterol. The liver and intestines are the major areas of cholesterol synthesis. The liver is going to produce about 1,000 milligrams of cholesterol per day, and the normal or average blood levels of blood cholesterol is 180 to 200 milligrams per deciliter. This always bugs me. This should be a capital L. Deciliter. There. We'll want to remember that the liver is the main producer of cholesterol. A lot of this will tie back to the liver. A lot of the fatty processes tie back to the liver. This is actually why the liver is involved in a lot of different processes and different important roles within the body. We talked about the liver when we talked about blood sugar, dealing with blood sugar and controlling our blood sugar and everything. We talked about it with detoxification of the blood and cleaning the blood. We talked about it it's also going to be vital in helping with our digestion and that kind of stuff as well. Working together with the gallbladder to produce all of the different enzymes and stuff that we need. And this is actually why the liver is one of the most important organs in our bodies. And in reality, that's actually why when we talk about cancers, somebody who has cancer that has metastasized into the liver, at that point it's typically no longer operable, and there's nothing that really can be done, because removing the entire liver and also just how vascularized the liver is, and it being such a vital organ to all these different processes, really can't do much after cancer or a disease process has gone into the liver. On the other side, the liver is also very, very cool in that it can regenerate itself. So you can actually donate a portion of your liver to somebody, and your liver will regenerate by itself, which is really cool. Okay, so cholesterol packaging and transport. Endogenously synthesized cholesterol. So endogenously, we're talking about endo, so it's inside. So it's cholesterol that's synthesized inside of the body. And dietary cholesterols are esterified. So they're the cholesterol ester. We looked at that type of bond formation, and that is the storage form of cholesterol. So the cholesterol is packaged into these very low density lipoproteins. We'll call those VLDLs in the liver. That liver, again, is the cholesterol synthesizing tissue. The cholesterol will also be packaged into these proteins called chylomicrons in the intestine, and that is another cholesterol synthesizing in tissue, the intestines. Although typically, I think this might be a typo. This is more referring to the dietary cholesterol within the intestine. When we're looking more at the dietary cholesterol being absorbed within the intestines. And those get packed into chylomicrons and then transported through the bloodstream. Okay, so we talk about a VLDL. We'll start to go look at some other examples as well. But this is kind of what we're talking about. We have these proteins. They're called lipoproteins. Lipoproteins, it's a combination of lipid and protein. And so you're going to have the protein and the fats all bundled together. And that's how they're going to be transported. They'll be held in the liver, and they'll actually be sent out into the body to drop off the cholesterol wherever the cholesterol needs to be. And then that's as it drops off cholesterol, the density of that lipoprotein is going to change. And then we'll see the introduction of LDLs and HDLs and stuff. We'll get into those a little bit. Just actually right next. So cholesterol is going to be transported by lipoproteins. So proteins combined with lipids. We have the very low density lipoprotein, the low density lipoprotein, and high density lipoprotein. So VLDLs are holding lots of cholesterol. The low density lipoproteins are holding less cholesterol than the very low density, and the high density lipoproteins are holding very little cholesterol. When they draw blood, this is actually the kind of things that these are what they're looking at. They're looking at your VLDLs, mostly your LDL and your HDL. So if you have high HDL, that's good. That's what we want. Having having a lot of high density lipoproteins means that these VLDLs that are leaving the liver are dropping off the cholesterol to the cells and everything that need them, so that there's not an excess amount of cholesterol. If you have a high concentration of LDLs, those low density lipoprotein, that means you're going to have excessive cholesterol, and that's kind of what they look for in order to tell if you have high or low cholesterol, and kind of what level of cholesterol and what risk factors that entails. This you'll talk quite a bit more about that as well in physiology. Physiology 2, you'll talk more about that. So cholesterol transport and its removal from blood circulation is controlled mainly by low-density lipoproteins and receptors on the liver cell membrane, and thereby depends on the LDL receptor concentration and the LDL receptor synthesis. Peripheral tissues take up LDLs, which are resulting from VLDLs, by receptor-mediated endocytosis. So receptor-mediated means that there's a receptor that needs to be involved. Endocytosis, the cytosis is kind of like swallowing or absorbing or taking in. Endocytosis is bringing it inside of the cell. Then the acyl groups of cholesterol esters are removed by hydrolysis, and then free cholesterol is released inside of the cells. Talking about the fate of cholesterol, the following dietary intake of cholesterol and synthesis of cholesterol, it may be packaged into chylomicrons in the intestine. That's the dietary cholesterol. And in VLDLs, this is the biosynthesized cholesterol in the liver. The chylomicrons are converted to chylomicron remnants by losing lipids, particularly triacylglycerols. So you're talking about it's in the form of triglycerides, or AKA triacylglycerols. Those triglycerides get packed into the chylomicrons. The chylomicrons are later, as it goes and it's dropping those off, it's converted to chylomicron remnants as it loses those lipids. For the VLDLs, as it passes out cholesterol, VLDL gets converted to an IDL, which is an intermediate density lipoprotein, LDL, which is a low density lipoprotein, and an HDL, which is a high density lipoprotein. And we say the density is looking at is how much fat there is versus protein. The proteins are very, very dense, adding all of the fats makes it less dense. That's why so high cholesterol to low cholesterol. You'll get asked a couple of questions on these, specifically some simple questions like which of the following has the most cholesterol in it, or which of the following is considered the good cholesterol. HDLs are the good cholesterol. VLDLs have the most cholesterol of them. Continuing with the fate of cholesterol, it can be converted to bile acids. It can be converted to cholesterol esters, so it can go back to the storage form. Serve as membrane lipid constituents. So we're going to use the lipids to make our membranes. It can be re-esterified as intracellular storage molecules. It can be transported to the liver by an HDL. And it can be converted to a steroid hormone. That's another thing about the HDLs. That the HDLs floating around in your blood, they're going to kind of be like the cleanup crew. They're going to come by and mop up and soak up any additional cholesterol that is not going to be used by the body and transport it back to the liver. So that's why we want those guys hanging around, so that way they can take all that unneeded cholesterol, so then the cholesterol doesn't end up sitting somewhere where it's not supposed to, like an artery or something like that, to prevent disease. Okay, so the biosynthesis of cholesterol. So cholesterol biosynthesis occurs in the cytosol of the cells. It needs hydrolysis of high energy, so thioester bonds of acetyl-CoA, and phosphoanhydride bond of ATP. So we're going to be using ATP. The acetyl-CoA or the acetate. Acetyl-CoA or acetate is the source of all 27 carbon atoms of cholesterol. So we have 27 carbon atoms of cholesterol. The sources of that acetyl-CoA or acetate is the beta oxidation of fatty acids, as well as oxidation of ketogenic amino acids, leucine and lysine specifically. I believe there's another ketogenic amino acid. Let me real quick pull up my board notes because I know that it says it in there. Lucy lies to get the keys. Okay, it is just leucine and lysine are our two ketogenic. There's our mnemonic to remember that, though. Lucy lies to get the keys. I want you to remember that for boards. As well as through the pyruvate dehydrogenase reactions. If you think about when does the pyruvate dehydrogenase reactions occur, that is happening at the end of glycolysis. After we produce pyruvate, we go have the pyruvate dehydrogenase complex. The 3 or 4 different parts of that complex break down the pyruvate into acetyl-CoA. The sources of reducing equivalent is NADPH, and we'll also have glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase of the pentose phosphate pathway catalyze the biosynthesis of NADPH required for cholesterol biosynthesis. So that, again, bringing up that pentose phosphate pathway, and we need that NADPH for this whole process to occur. Again, this is going to be a very similar process to what we saw earlier with the traditional lipid biosynthesis in Chapter 2. Okay, so the major stages of cholesterol synthesis are as follows. This is where we're going to get kind of in the meat of stuff, where again, you'll want to write out this reaction, write it in a way that makes sense to you, condense it down a little bit, just to make sure you remember the different intermediates, especially the enzymes, and work through those. Again, now that, from my understanding, all tests will be in person, So this is something that you definitely need to really, really know and know well, and be able to when you get your paper exam, you can have a piece of scratch paper, use one of the pages as a scratch paper to be able to draw out this process quickly. So if you can come up with mnemonics or abbreviations or stuff for it, I highly recommend that as well. Okay. First off, we have the condensation of 3 acetyl units and formation of mevalonate. 2 molecules of acetyl-CoA condense to form acetoacetyl-CoA by thiolase. So our goal is to get here to mevalonate. We have an acetyl-CoA. We have our thiolase. We have our HMG-CoA synthase. Breaking down and using the HMG-CoA reductase with the NADPH, we get to mevalonate. And this is most of this should be review from last chapter, from chapter 2. From mevalonate, we have the mevalonate 5-phosphotransferase, using the ATP to take a phosphate, putting that in there at the end. Again, then a phosphomevalonate kinase. Remember, the kinases are going to also phosphorylate. So it's going to add another phosphate group. And then pyrophosphomevalonate decarboxylase. We're going to be adding this another phosphate group onto here as well. Then we have pyrophosphomevalonate decarboxylase. Then we are getting down here, we have activated isoprenes. Let me find you a good definition of isoprene, as that's a word that's going to keep getting used. So the isoprene, I want to look right here. Isoprenes are used, the isoprene units are used in biosynthesis of naturally occurring what are called isoprenoids, such as carotenoids, quinones, lanosterol derivatives, for example, steroids, right here. So isoprenes are a specific chemical compound here within this process that we're going to be using eventually, and we can use them later to eventually as the building blocks to continue to build cholesterol and then get to steroids and those other things that we need. Let's see if we have if it gives us the actual, yeah, it gives us the actual step-by-step here. It would probably be good to take some time and match what this is saying with the different steps that we see above, in order to give us a better idea of exactly what is taking place. So here, the third molecule of acetyl-CoA condenses with acetoacetyl-CoA to produce beta-hydroxymethyl-gluteryl-CoA, and then also known as HMG-CoA by HMG-CoA synthase. So that right there is talking about right here. It becomes HMG-CoA. The HMG-CoA is converted to mevalonate by HMG-CoA reductase. The HMG-CoA reductase requires 2 molecules of NADPH. So these reactions, and the HMG-CoA reductase is the rate-limiting enzyme in cholesterol biosynthesis. So we always want to remember that the HMG we always want to remember what the rate limiting enzyme or what the rate limiting step is, because that is the most likely thing to get questioned. Step 2, we have the activation of mevalonate. So we have mevalonate plus ATP. It's the mevalonate with the phosphorus, and then there's going to be another ATP mevalonate with 2 phosphates. And that's what we saw right here. The mevalonate gets 1 phosphate group added, and then another phosphate group added, and those are those specific 2 steps that we're looking at. Now, so the conversion of activated mevalonate pyrophosphate to activated isoprene units. So we talked about what isoprene units are. Those isoprene units, it's just the name for the chemical structures that are going to be at this stage. They're volatile hydrocarbons that are going to be used to eventually create our cholesterol. So in the next 4 stages, mevalonate is activated by phosphorylation by 4 different enzymes and converted to form activated isoprene unit, also called isopentenyl pyrophosphate. So our mevalonate double phosphated gets converted to isopentenyl double P, and that is also in between the dimethylallyl diphosphate with the pyrophosphate, our activated isoprene with pyrophosphate. So these are the 2 names that you want to remember, the isopentenyl and the dimethylallyl. And that is these steps right here. So you have the 1, 2, 3 ATPs are being used. And finally getting to, we have a decarboxylase reaction, and then we finally get our activated isoprenes, that isopentyl and the dimethylallyl pyrophosphate. Next, polymerization of 6 5-carbon isoprene units to form 30-carbon linear squalene. So what this means, polymerization, means that they're going to be joining together. This might age me a little bit, but when I was a kid, I used to watch Yu-Gi-Oh! And there was a specific card that was polymerization. So, and that what that did is it fused different of the Yu-Gi-Oh! Monsters together. So, if at any point you've watched that show, or if you want to go a little thing to keep in your mind, go look up that clip, look up a clip from that, and maybe that'll help you remember that. Okay, but the polymerization is the combination of 6 5-carbon isoprene units. We're going to take those activated isoprenes, and now we're going to stick 5 of them together to form a 30-carbon linear squalene. This is important to note. Squalene is a 30-carbon linear structure. And if we think, though, cholesterol is a ring structure, so in a little bit we're going to get to more of that ring structure. And then this is going to go back over the steps. So first, 2 isoprene units join and produce a 10-carbon geranyl pyrophosphate. Geranyl pyrophosphate reacts with another isoprene unit and produces the 15-carbon farnesyl pyrophosphate. And besides producing squalene, farnesyl pyrophosphate also can be used to generate other important isoprenoid compounds like dolichol and ubiquinone. Ubiquinone, if we remember, ubiquinone it's also called coenzyme Q. And if you remember, where did we see coenzyme Q, or CoQ10 as well is another name for it? That's in the electron transport chain. Dolicol. I don't know what dolicol does. Dolicol function. That plays a role in the co-translational modification of proteins known as N-glycosylation in the form of dolicol phosphate. So it helps with modification of proteins. Also an important function. That helps with the electron transport chain. That farnesyl pyrophosphate. So then we have, so we're going to be building, essentially building 2 of those. So here we put 2 of them together, get adrenal pyrophosphate. We have 2, and then we're going to add in another one to get our farnesyl pyrophosphate. And then we have a second farnesyl pyrophosphate, and we're adding those together, so 15 times 15 to form a squalene. And if you notice and you look at this, this kind of looks like you could just sort of fold it over and kind of connect the 2 and get those nice ring structures. So that's kind of pretty much exactly what it's going to end up doing. It's going to kind of fold and start to form those ring structures, but we'll get to that in one more sec. So the 2 of the 15 carbon farnesyl pyrophosphates combine and form the 30 carbon squalene. Thus, condensation of 6 isoprene units forms squalene, a linear 30 carbon compound, non-cyclical compound. Right here, those 6 of those 5 carbon isoprene units. Okay, now that we have our squalene, cyclization of squalene to form the 4 rings of the steroid nucleus with a further series of changes, oxidation, and removal or migration of methyl groups to form cholesterol. Squalene is oxidized and cyclized to produce lanosterol. Let me actually see if I can find a good example of... So just your cyclization reaction specifically is the formation of a ring in a chemical compound. So cyclization of squalene, that's literally saying that it's forming it into the ring structure. So the squalene is oxidized and cyclized to produce the lanosterol. So we can see that here, squalene starts to get acted on, does all of its stuff. Where is our cyclase? There's our cyclase, and now we end up with lanosterol. And then further modification and removal of 3 carbon units produces our ending cholesterol, which is 27 carbon atoms. There are use of inhibitors of some enzymes in the cholesterol synthesis pathway is what we're going to see in this next bullet point down here. So squalene monooxygenase inhibitors could be a target for hypercholesterolemic therapy. You'll talk more about this study in class, but basically this is talking about how the squalene, like the different enzymes and stuff that are making and producing the squalene in order to produce cholesterol, we can target those using drugs in order to actually stop the body's production of cholesterol in somebody who already has hypercholesterolism or who has high cholesterol already. Because the typical medication that we would use are called statins. Statin are the agents, the medications that we use to help control high cholesterol. But besides statins, now non-statin agents are also now considered. So potential target enzymes are the squalene synthase, the farnesyl diphosphate farnesyl transferase, the squalene epoxidase, and the oxidosqualene cyclase lanasterol synthase. Kind of those two going together. I would recommend for his exams knowing a handful of these, specifically the squalene synthase. I remember that he likes to ask questions specifically about that, and also make sure that you know what a statin versus a non-statin agent is. Okay, so the regulation of cholesterol biosynthesis. We have a rate-limiting step. That HMG-CoA reductase is that rate-limiting enzyme with a rate-limiting step. It catalyzes the rate-limiting conversion of HMG-CoA to mevalonate. The fasting condition decreases HMG-CoA reductase activity. So if we are fasting condition, we are decreasing that reductase activity, which then in turn decreases cholesterol synthesis. Because if we don't have the components, the extra components to be making cholesterol, then then our body's not going to do it. And that is controlled by a negative feedback mechanism where cholesterol directly inhibits its own synthesis. So once we have enough cholesterol, or a decent amount of cholesterol in our system, the cholesterol is going to be sensed, and it's going to come back all the way to the beginning and turn off the HMG-CoA reductase to prevent more cholesterol from being produced. HMG-CoA reductase is inactivated by phosphorylation, which is catalyzed by AMP-dependent kinase. So certain things such as glucagon, cholesterol, or glucocorticoids will shut it off. It is activated by dephosphorylation. Through the activators are insulin and thyroid hormones. And remember, thyroid hormones are ones that are going to be helping to control our metabolism as well, and as well as our heat production and a couple of other roles in the body. You'll talk even more about the glucagon, the glucocorticoids, insulin, and thyroid hormones a fair bit in physiology too, and then a decent amount as well in systems pathology in Tri 4. Production of HMG-CoA is regulated at 3 different levels. Transcriptional control, so the role of sterile regulatory element binding proteins, SREBP, and SREBP cleavage activating protein, or SCAP. So we're talking about the actual making of HMG-CoA being regulated at the 3 different levels. So it can be, let me see if there's... Okay, so transcriptional control, we can see here it can literally be at the gene level, controlling it at that specific to prevent the production and the making of the actual proteins, where the sterols will inhibit the production. B, the proteolytic degradation of HMG-CoA reductase, here seen in B, where the sterols, when we have plenty of sterols, it's going to encourage the proteolysis degradation and breaking that down. And also regulation by covalent modification, that phosphorylation and dephosphorylation reactions, where the AMP activated protein kinase with ATP, and then the different glucagon sterols are going to activate the protein kinase, which will then phosphorylate it to inactivate it, or dephosphorylate it to reactivate it. And this chart, this is just going over what parts insulin and glucagon they play, and like where those negative feedback loops are. So ideas to lower cholesterol levels, the use of inhibitors of some enzymes in the cholesterol synthesis pathway. This is just a repeat that we saw up above, repeated here. Little clinical correlates. Cholesterol lowering drugs. So statins are competitive inhibitors of HMG-CoA reductase enzyme. So rather than the HMG-CoA reductase, rather than the HMG-CoA getting bound to the reductase, they can get bound to the statins, and then it prevents them from going through the process of continuing to become cholesterol. We have examples of the statin compounds right there. I don't remember him asking any questions about that, but just read through the list once or twice, just to have especially right before the exam. Put a little star here just to read through it, just so that way, if he does ask a question, you have a better chance of recognizing one of the names. And these statin compounds resemble mevalonate. Okay, so besides statins, non-statin agents are now considered. This is a repeat we looked at up above. Atherosclerosis. So deposition of cholesterol and cholesterol esters occurs in the connective tissue of the arterial wall. It is characterized by the elevated levels of VLDL, IDL, and LDL as seen in diseases such as diabetes mellitus, hypothyroidism, hyperlipidemia, etc. You'll also see really atherosclerosis. Once you have that plaque, that buildup of the fats and stuff in the lungs, it contributes also to CVD, cardiovascular disease. A quick note here, diabetes mellitus. I want to make this super so. Diabetes mellitus is insulin issues. Diabetes insipidus, which you'll learn about next trimester, a handful about, and in a couple of other classes as well, again in physiology and systems pathology. Diabetes insipidus is a pituitary gland issue, okay? And that's going to be it's called a water control issue, where your body has trouble regulating the water supply inside of the body. Let me see, how long is this bile part? Okay, we'll get through this bile part. This is, I feel like, the part that a lot of people get really tripped up on. I'm talking about the bile acids and the bile salts. So I'll try to break this down and make it pretty clear. So the nature of the bile, it is an amphiphilic molecules, or a biological detergent. All this means, detergent, means that it breaks down fat. It breaks stuff down, it pulls it apart. It's designed, and its purpose is to break down fat. They are going to be produced in the liver and secreted into the bile. Okay, so they are produced in the liver, and the bile is produced, and then it gets excreted and stored in the gallbladder. Okay? So the functions, they are released into the intestine during a meal. It helps with digestion of dietary lipids. They make the dietary lipids soluble by producing micelles. And thus, these micelle-containing lipids become available to intestinal lipases for breakdown. Let me quick pull up. So micelles are temporary compounds formed during the fat digestion and absorption process. They are water soluble and enable the lipid digestion products to be transported to the small intestine surface for absorption. Let's look at some pictures. So this is kind of what you can imagine a micelle looks like, where it's got all the fats stuff it forms and makes it water soluble. Remember from last try, like dissolves like, where if a fat typically is not going to be water soluble. So by being packaged in these little micelles, it makes them water soluble enough to transfer them across the membrane in order to get them through the small intestine and into the bloodstream, and into to be able to get them to where they need to be transported into the actual rest of the body. Micelle-containing bile acids also take up lipid-soluble vitamins and the product of lipid digestion. If you remember, we talked about the vitamins. Vitamin A, D, E, and K are our lipid-soluble vitamins. There are two sets of primary bile salts that are formed from cholesterol: cholate or cholic acid, and quinocholate or quinocholic acid. Okay, these are our bile salts: cholate or cholic acid, quinocholate or quinocholic acid. 7-alpha hydroxylase is the rate-limiting enzyme in primary bile salt production. This enzyme adds an OH group at the 7th carbon in the cholesterol molecule. This enzyme is feedback inhibited by its end product, primary bile salts. This enzyme is also regulated by phosphorylation and dephosphorylation mechanism, in contrast to the HMG-CoA reductase, which is the rate-limiting enzyme in the cholesterol biosynthesis. This enzyme, the 7-alpha-dehydroxylase, is activated by phosphorylation. So this HMG-CoA reductase is deactivated by phosphorylation. The 7-alpha-dehydroxylase, so the production of the primary bile salts, is activated by phosphorylation. And that gives us our these are our primary bile salts. Our secondary bile salts, deoxycholic acid and deoxyquinocholic acid, are produced by removal of one hydroxyl group in an oxidative matter. Okay, see how we have our hydroxyl groups. Now we're missing that one right over there. And that is our secondary bile salts. And so this is actually called, this is what I want to make sure that I throw in here is, because this isn't really explained in the book. So the primary bile salts undergo that dehydroxylation by bacteria in the small intestine, forming the secondary bile acids, that deoxycholic acid, and it's called lithocholic acid. It's the same thing as the quinocholic acid. And that's how we get the secondary bile salts. It's due to the bacteria in the small intestine. The bacteria in the small intestine are responsible for that. That just helps make more sense in my brain. So if we think, if the bile salts are sitting in the gallbladder, what type of bile salts do we have? We have primary bile salts. Once they're released into the small intestine, after they've finished helping with digestion, what do we have? Then we have, when they've been helping with digestion, they're now secondary bile salts. And then we also have something called conjugated bile salts. So the conjugation, when we're talking about conjugation is meaning combination, like combined. So it's the combining of bile salts with the amino acids, specifically taurine and glycine, which helps to make them better detergents. Let me actually show you a specific define detergent in chemistry. So detergents, increasing the spreading and wetting ability of water by reducing its surface tension, the affinity its molecules have for each other, in preference to the molecules of the material being washed. So typically, the water would never want to associate with the fats, but thanks to the detergents, the water is then able to interact with the different fats. Okay, so they interact with the taurine and the glycine. So cholic acid plus taurine becomes taurocholic acid. Cholic acid plus glycine is glycocholic acid. Conjugation of bile salts with taurine and glycine also lowers the pKa value from pK6 to pK2 and pK4 respectively. So with taurine, it lowers it to pK2 and glycine to pK4. And that makes them more acidic, soluble, and absorbable when combined with lipids. Deconjugation may be caused by some intestinal bacteria, which make them less soluble. Some clinical correlates. So cholesterol gallstones can be formed from cholesterol. This is why some individuals, if you're doing like the keto diet, high protein, or like a really high protein diet for a long period of time, you can get gallstones, as well as kidney stones from doing something like that. Quinodeoxycholate or ursodeoxycholate is used to prevent the development of cholesterol gallstones by dissolving gallstones. This medicine is a hydrophilic bile salt that reduces cholesterol content in bile. Lecithin cholesterol acyltransferase, LCAT, deficiency causes an inability to form cholesterol esters from HDL-associated cholesterol. This results in a decreased ability to remove blood cholesterol and manifests as defects in the kidneys, red blood cells, and the cornea of the eyes. And then here we just have a brief outline of synthesis of steroid hormones from the cholesterol. Cholesterol gets converted to pregnenolone, pregnenolone to progesterone. Progesterone can be turned into cortisol or testosterone. Progesterone can also go to corticosteroid or mineralocorticoid, which then becomes aldosterone, which is a mineralocorticoid. You'll learn a lot more about these and like specifically where these different steroid hormones come from next trimester in Physiology 2. Okay, so that is all of Chapter 3. This video is a little bit longer. Glad that we were able to get through all of it and still kept it under an hour. So good luck studying, everybody, and hopefully you learned a couple of things from this video.